Hi and welcome to a quick explainer video on translation. Okay, so first we need to consider the coding problem. We have 4 nucleotides in messenger RNA, G, A, C and U, and we have 20 amino acids. So therefore we can't do a direct one-to-one swap between messenger RNA and the protein sequence as there are not enough bases. Therefore we must use a combination of bases to encode for each of the different amino acids in the protein. So 1 nucleotide is not enough. How about 2? Well, that would only give us 4 to the power of 2 or 16 base combinations. Again, not enough. How about 3? Well, that gives us 4 to the power of 3, which is 64, and that is too many. So we can't use 2 as that is too few, and as 3 is more than enough, then the cell uses 3 bases to cover the 20 different amino acids that can be used to make a protein. And we refer to these triplets of bases as codons. As we have seen, we have more potential codons than we need. However, the cell uses all 64 codons because it uses more than 1 codon for each amino acid. For some amino acids, the cell uses 1 codon, for some 2, for some 3, for some 4, and 3 amino acids have 6 codons. In a number of cases, for example alanine, the cell uses GC for the first 2 letters of the codon and then any of the bases in the 3rd position. The 3rd position is often referred to as the wobble position, as for a number of amino acids it doesn't really matter which of the bases occupies that position. Now, the use of multiple codons for most amino acids does cause us an interesting problem, particularly in the lab, and that is the fact that the code is degenerate. That is, we can accurately predict the sequence of amino acids from a string of codons, but we can't predict the string of codons from an amino acid sequence, as we may have more than 1 codon for each amino acid. And this is referred to as degeneracy. And there is also a problem with trying to predict the amino acid sequence from a given DNA sequence, because we need to know the correct codons to translate. So what do I mean? Well, take this simple stretch of 14 bases. Where are the codons? We could start at the first base C and then do 3s like this. Then we get the amino acids leucine, serine, valine and threonine. However, we could start the codons here at the U, in which case we get different codons, which when translated give us serine, alanine, leucine and proline. Finally, we could move in 2 bases and start at the 3rd base. This gives us a different set of codons. And when we translate them, we get glutamine, arginine, tyrosine and histidine. So that gives us 3 potential reading frames per strand of messenger RNA. But it gets worse. As we are assuming for a given piece of DNA, we know which of the 2 strands gives the messenger RNA. In fact, for a given DNA sequence, you have 2 potential messenger RNA molecules, and each of those can produce 3 reading frames. So this gives us a total of 6 possible reading frames for 1 piece of DNA. Therefore, there is a bit of a problem. How do you identify the correct reading frame that gives you the protein? Or to give its proper name, how do you determine the open reading frame in the DNA molecule that gives you the correct protein? The codon sequence just doesn't start straight after the 5' cap on the messenger RNA, but starts some distance away. That is, there is what we call a 5' untranslated or UTR region. There is also one at the 3' end, between where the protein finishes and the poly A tail. The 5' UTR contains the binding sequence for the translation machinery and also helps the machinery determine the correct reading frame in the messenger RNA. In this process, one thing that is always true is that the first codon in the messenger RNA sequence is an AUG, which is referred to as the start codon and gives a methionine. Further downstream in the messenger RNA, you will find in the same reading frame one of 3 stop codons, TGA, TAA or TAG, that tells the translation machinery to stop making a protein. Past the stop codon and before the poly A tail, you will find the 3' UTR. However, getting back to the protein, Just because the start codon always gives you a methionine, then it doesn't mean that every protein in the cell starts with a methionine, as the N-terminal end of a protein is usually processed and the methionine removed to make the final mature protein. So how does the cell actually make the protein? Well, the amino acids do not directly bind to the codons, but instead bind to a transfer RNA that contains an anti-codon to the codon in the messenger RNA. The amino acid is matched to its transfer RNA by an enzyme called aminoacyl-tRNA synthase, and there is one of these enzymes for each of the potential transfer RNA amino acid pairs in the cell. The process of translation starts in the cytosol of the cell with the binding of the ribosome to the messenger RNA. The ribosome can be viewed as a complex protein synthesis machine made up of more than 80 ribosomal proteins and several ribosomal RNAs. The ribosome consists of two subunits, the large subunit and the small subunit. Each ribosome has 4 binding sites, 1 site for the messenger RNA and 3 for the transfer RNAs. The 3 tRNA sites are called the A site, the aminoacyl site, the P site, the peptidyl site, and the E site, the exit site. The ribosome works like a little machine. A tRNA will only bind to the A and P sites if their anticodons match the codon on the messenger RNA. Having both sites occupied means that the reading frame is maintained. The process of growing the peptide chain can be split into 4 steps. Step 1, the incoming tRNA with its bound amino acid binds to the A site by base pairing with the messenger RNA codon. Step 2, the carboxyl end of the growing polypeptide is released from the tRNA at the P site and joined to the free amino group of the amino acid at the A site. This forms a new peptide bond. Step 3, a conformational change in the ribosome shifts the 2 tRNAs into the P and the E sites, moving the messenger RNA exactly 3 nucleotides, 1 codon, through the ribosome. Step 4, another series of conformational changes in the ribosome results in the release of the tRNA at the E site and resets the ribosome in preparation for the next tRNA amino acid. And then it's back to step 1. And this process will be repeated until a stop codon is encountered. Now, the ribosome can add about 2 amino acids per second, and this means an average protein of about 500 amino acids would take about 250 seconds to make, or roughly 4 minutes. So, to speed up this production process, the cell will often have multiple ribosomes running down one messenger RNA molecule, and these are called polyribosomes or polysomes and can be seen using an electron microscope. The final stage of producing a functional protein from the messenger RNA sequence is to get the protein to correctly fold so it works, and if needed, modified and packaged so it ends up in the right part of the cell. The code that says how a protein should fold is encoded in the chemistry of the amino acids that make up that actual protein. This folding can also be helped by a group of proteins called chaperones. In terms of getting the protein to the right place in the cell and, if needed, correctly modified, this is also encoded in the string of amino acids. Okay, so that's it for this video on translation.